Okay, hi everyone. Happy Friday to you. So today we'll wrap up our discussion about the challenges that our immune system presents when it comes to transplanting tissues in modern medicine, right, from donors to patients to recipients. So let's finish talking about these grafts and transplantation issues. So let's quickly review so that we all are on the same page of where we left off last time. We had been discussing that um, the types of tissue rejection that we have to deal with when you're talking about donors and recipients, these follow the same types of hypersensitivity models as do allergies. In particular, type 2, type 3, and type 4 hypersensitivity models are what we encounter with grafts and transplantations. Remember, type 2 hypersensitivity is when you have a patient has existing IgGs against something like the wrong ABO blood type. That can lead to very quick, sudden rejection of things like the wrong blood type. If you kind of screw up the ABO blood type matching during a blood transfusion, because remember, blood is technically a tissue as well, right? And importantly, if you're talking about solid tissue transplantation, because solid tissues are filled with blood vessels, right? And not only would there be blood cells in there, but also the walls, the endothelial cells of things like capillaries also express ABO antigens on them. Then that's another way that if you mess up the ABO blood type matching, you can not only lead to rejection of the wrong type of blood being transfused, but also of solid tissue being rejected when transplanted. And that can happen very quickly, oftentimes within just a few hours. Right? So we called that hyperacute, a very rapid onset. We said that type 4, moving down to type 4 here, involves activation of T cells against the wrong HLA type. Remember last time we discussed that your HLA polymorphisms that you inherited from mom and dad that give your MHC platforms on the surfaces of your cells, slight different variations in shape. That's great for us as a species because the more diverse sort of selection of different shapes of these platforms that different people can make on the cells in their body, that means at least some people will have better shapes for being able to present different small peptides from things like pathogens. Right? So that's why some people's bodies would be more responsive. Their immune systems would have an easier time of detecting the presence of this pathogen because the cells in their body are doing a better job of presenting peptides from that pathogen because of the particular HLA polymorphisms that determine the shape of the MHC platforms, right? But in terms of when we're talking about tissue transplantation, this is a big challenge, right? Because everybody has diverse polymorphisms and everybody has slightly different shaped MHC proteins, right? That means it's very hard to expect that a big sort of chunk of tissue, like a complete organ, like a kidney, right, that's transplanted over, right, would be tolerated well by the patient's immune system that knows to expect self-HLA because that's how it's trained, right? But it would never have the reason to tolerate or be okay with a foreign sort of HLA type, right? And so that can lead to rejection of the tissue within days or weeks following transplantation. And then type 3 is more of a chronic, drawn out process. That has to do with if, for example, the HLA matching was pretty good, right? So they're able to not only match the ABO blood type between donor and recipient. I mean, that part is trivial, right? Doing the ABO blood type matching is pretty trivial. But getting HLA matches is harder. right? So that's why most of the time donors involved in a organ transplantation are usually like a family member, a close family member, because genetically they're closer. So that means there's a greater chance that their sets of HLA polymorphisms are more compatible. Right? But even then, over time, because there's going to be slight differences, right, and the immune system is so sensitive and geared up to do its job of identifying things that are different, things that are not self, right? Then over months and years, you can get a situation where eventually the patient's immune system has kind of geared up and begun selecting for things like B cells and T cells that ultimately lead to chronic kind of damage being done to the donated tissue. And that's why I said at the 10-year mark, about 40% of all solid tissue transplantations end up failing, right? Okay, so here's a question. Why are things like the ABO blood typing, the RH plus minus status, and HLA polymorphisms, why are these so immunogenic, right? So this term immunogenicity has to do with how easily they actually cause problems with the immune system. Consider this. The majority of antigens that come over as part of a tissue graft, meaning all of the cells that make up something like the kidney that's being transplanted, most of those antigens are nearly identical to the patient's own self-antigens, right? In other words, the kidney cells are perfectly healthy, normal human kidney cells, and the proteins that they make as part of normal cellular function, many of, if not most of them, would have little variation between the version that the donor has and the version that the recipient has, right? Because most of the proteins that are encoded for by our genes are not highly polymorphic. Right? If evolution got us to this point after hundreds of years of evolving into human beings, right, and our human versions of genes that code for different proteins are the ones that have been sort of like finely tuned and kind of worked out, right? Then you would imagine that, for example, the keratin that makes your skin or your hair is not going to be really all that different than the keratin gene that makes my skin or my hair, right? So you wouldn't expect many of the different types of antigens in healthy tissue to be the cause of tissue rejection. Right? But of course, the exceptions are these ABO and HLA types. I said they're highly immunogenic, meaning that they are really strong signals that immune systems pick up on very easily. And when you have an immune system activating against something that's actually normal tissue, there's nothing wrong with it, right? It's a perfectly healthy kidney that's being donated over. Again, we call that aloe immunity. Aloe simply meaning it's the same thing. It is a human kidney, right? But aloe means, but it's from a different source, meaning it came from somebody else. And the reason for that, the reason why ABO and HLA types make such a big difference is because, number one, think about this. They are antigens that are located on the cell membrane, right? So they're not proteins that are kind of doing stuff inside of a cell. They are meant to be on the outside of the cell. Therefore, they are meant to be visible to things like the immune system. So that makes them easier to detect by our immune system, right? Consider in the case of things like ABO blood types or RH plus minus blood types, patients already have elevated IgG titers for things like the wrong AB blood type, right? Again, we discussed this. It's because those ABO antigens are glycolipids that, resemble in many ways the lipopolysaccharides that we find on the surfaces of bacteria. And after eons of co-evolving with bacteria, right, our immune systems have become preset, right, pre-programmed to be very sensitive to being able to detect things like lipopolysaccharide signals from bacteria that's infecting our tissues. So therefore, that's why it's almost a given that you will have IgG in your system already against other ABO blood types, simply because there's no reason for your immune system to have ever uh, been tolerant, right, or accepting of a different ABO blood type, and so therefore you probably have IgGs already, right? And lastly, because HLA proteins, right, make those MHC platforms, right? So the MHC platforms, remember, are made of HLA proteins, and those are the very proteins that our T cells are designed to kind of look for, right, and interact with their T cell receptors, right? This combination of these three sort of issues here is the reason why you end up with so many problems having to do with wrong blood type and HLA mismatches. So let's explore this in a little bit more depth. Right? Remember, we said that the involvement of T cells and B cells in rejecting tissue can happen either pretty fast in a direct or acute kind of way, right? Or it can be happening in a more drawn out kind of way. This is an example here where, for example, a kidney has been transplanted. So this is the kidney from the donor. It is then surgically hooked up or put into place inside the patient's body, right? And then as a result of the HLA matching not being, too close, as a result, what happens? Here you would have a donor's dendritic cell that may be like hitched a ride, for example, in the donated kidney. Then you would have the host's own naive T cells, whether they're helper T cells or cytotoxic T cells. They just recognize that this MHC type on this foreign tissue is wrong, right? It's not self HLA, right? It is some other foreign HLA. And again, because these T cells in the host have only been trained to be okay with self HLA, right? Therefore, naive T cells that happen to have the right shaped TCR to bind by chance to a foreign shaped HLA, those cells never would have been screened out of your system, right? Because why would they have been? Right? Because there's no reason why those T cells shouldn't be allowed to exist. Again, because Mother Nature never intended us to put somebody else's kidney, right, in your body, right? So in this case, then, the activated T cells would very quickly just respond and freak out by binding to the donor's MHC type. That would lead then to activation of cytotoxic cell activity as the CD8 T cells begin to not only kill off the donor's dendritic cells, but then as they're circulating around in the body, they're encountering the new kidney, right? And so all the tissue of the new kidney ends up being attacked by the cytotoxic T cells as well. And within days, the graft is rejected, right? Okay. Chronic tissue rejection then would be a situation where, the physicians, the doctors, the surgeons have done a really good job of trying to match HLA types between donor and recipient, to the point where you don't get sort of direct rejection. You don't get acute rejection of the tissue because the recipient's T cells, are okay with the HLA on the donated kidney cells, right? Because it's close enough, because the HLA polymorphisms kind of match. That means the shapes of the MHC platforms are pretty close to what the host's own T cells have been trained to tolerate, right? I mean, another way of saying it really is that any brand new T cell that's forming in the thymus of the host, right, if it came up with a T cell receptor that reacts, strongly against this different shaped the self HLA, then that T cell would never be allowed to go to work, right? And so that's why only T cells that are tolerant of these shaped MHCs are allowed to go to work in your system. And therefore, if the doctors did a good job of matching HLA type, then the foreign donated tissues with their MHC platforms, look close enough to your own body's one so that your T cells are tolerant of it. Right? However, because if it's somebody else, there's going to be some genetic variations. So there are going to be slight differences in the shapes of these peptides that come from the MHC proteins. So over time, that's what causes a problem, right? So this is now a chronic situation drawn out over months or years. And in that case, right, we're dealing with indirect allo recognition. In other words, here is debris. This little white thing here is maybe a piece of the donated kidney that just naturally, right, cells get recycled all the time. Even if that healthy donated kidney, right, continues to be healthy in the donated body, in the recipient's body, right, it's still going to be constantly making new cells, and old cells are going to be dying and being recycled from it. So here's a piece of one of the old cells, right? And so it has the donor's MHC on it, right? After all, because this is from the donated kidney. So the recipient's immune system does its normal job. Here you have a dendritic cell that's scrounging around, and it scavenges, and it takes in these bits and pieces of the donated tissue. Of course, it's going to process the proteins that it finds on there, including the proteins that make up the HLAs that make up the MHC platforms. So it's going to present that yellow dot, right? So this yellow dot then does look kind of different than the yellow dot that normally you'd get if this were your own MHC type, right? So it loads it up on this MHC2. And over time, what you will find is there will be naive helper T cells that have been allowed to go to work, and they're just kind of going around between the blood and the lymph. And some of them might have the right shaped T cell receptor to recognize this yellow dot, as being something foreign, right? So this CD4 cell right here, this naive one, think about this. If this transplantation never happened, right, then this CD4 cell that was put to work going in your blood would never encounter this particular shaped yellow dot, right? So in reality, this CD4 cell never would have done anything, and it eventually would have been recycled. It would go away after, a few weeks, right, to be replaced by other naive T cells. But in this case, because there was a transplantation, it's actually able to see a peptide that its TCR recognizes. So as a result, this helper T cell will activate. You follow the blue arrow around here. Right? And in the lymph node, where it's seeing this, what else do you have in the lymph node? Well, we learned that's also where other immune cells get concentrated to try to encourage more immune cell antigen interaction. Right? And so that means you will have naive B cells in there. And this naive B cell, similar story, right? It came up with its own IG in the bone marrow as part of B cell development. It passed self-tolerance training, meaning it did not get into any trouble in the bone marrow. It did not get into any trouble in the blood. But now it encounters, right, this foreign HLA type that's different enough from self-HLA, right, that this B cell with its immunoglobulin that happens to be able to recognize it, right, is allowed to go ahead and activate. And as we said before, the real key event here in the lymph node that's happening is you have this B cell activating, you have this helper T cell activating, and they pair up, right? The B cell, once again, is processing the proteins that it's taking into the yellow dot, and it presents that yellow dot to that CD4 cell, right, that already was activated by the dendritic cell, right? So this B cell is like, hey, check this out. I found this yellow dot. Is this something that you're interested in, right? And then this CD4 helper cell is like, yeah, right, that is the yellow dot that my T cell receptor recognizes. And I've already been sort of woken up and alerted to this by the dendritic cell, right? So I've kind of been looking for you, right? Because we were meant to kind of be together here, right? And then this CD4 helper T cell then tells this B cell, all right, you start activating, make many copies of yourself, start the whole hypermutation process with some of you go ahead, turn into plasma cells and make now antibodies, right? That unfortunately can be thought of as being anti-HLA antibodies or allo antibodies, right? Because these antibodies are actually being selected for to recognize this slightly different version of a totally normal, healthy peptide. Right? So this process takes a long time. So it might take several years of building up levels of these different antibodies that are constantly then attacking the tissue, right? And after many years of damage, persistent damage, the kidney eventually fails, right? And the organ is rejected. All right. So it's one thing to say, okay, blood transfusion is important when maybe you have a patient who lost a lot of blood, right? So you need to hurry up and replenish the blood in their body. Otherwise, you're going to die because they've lost like a couple liters of blood or something like that, right? Or you say, well, we need to transplant a kidney because the patient's own kidneys have failed, right? Due to kidney disease, something or another, right? So now you need to put in a brand new kidney from a donor. In both of those cases, blood transfusions or organ grafts, you're talking about replacing something that's missing, right? Something that whose function suddenly is no longer there. Right? Stem cell transplants are different. The purpose of a stem cell transplant, I think you can already kind of reason this out, right? Stem cells give rise to all the other cells. Remember, in this class, we've talked a lot about the hematopoietic stem cells in your bone marrow that give rise to all the different types of cells that make up your immune system, that make up all the components, cellular components of your blood, right? So transplanting stem cells is more about reprogramming or kind of like rebooting an immune system that has run into trouble, right? Because the existing stem cells that the patient had, right, something's gone wrong with them, right, to cause disease. So now you're trying to replace those bad defective stem cells with new stem cells that can hopefully take over, right, and do the job of actually maintaining the healthy immune system and replenishing the healthy immune system that the patient's own immune system couldn't do. So these types of stem cell transplants are used to treat things like genetic diseases, right, where the person is born with particular mutations or allelic variations. That means their stem cells and their immune system is just kind of defective because of the genes that they have. So therefore, you can try to replace those stem cells, right, with stem cells that have genomes in them that are correct, right, or free of these bad mutations. Right? Also, stem cell transplants are used in cases of hematopoietic cancers. Right? If the stem cells of a patient's body are the source of their cancer, right, then what you can do is just say, well, let's just get rid of all those cancer-causing stem cells and replace them, right, with healthy stem cells. So right away, hopefully, you can sense that there are some additional challenges here when you're talking about a hematopoietic stem cell transplant, beyond simply things like ABO blood type matching or HLA type matching, right? Because you're trying to get rid of an old immune system and reboot it with a good immune system. So that means, number one, here are a set of 3 challenges, additional challenges. The recipient's old immune system first has to be eliminated, right? If the immune system itself is the cause of all the problems, right, you need to sort of like delete the corrupt immune system in order to replace it with a good copy, right? Like if you're talking about, like computer files on your computer, right? You have a bad copy of something, program or file or whatever, you need to delete it, right, before you can get a fresh copy put in. Otherwise, if you didn't first delete it, you would have 2 competing immune systems going at each other, right? And that type of allo reaction, I mean, that would obviously be intolerable, right? The other thing to think about is the transplanted graft that contains these hematopoietic stem cells. You better make sure that it is really enriched in these stem cells, right? Because if you're going to erase a patient's immune system, you better make sure that you have plenty of stem cells that you're bringing in to reboot it quickly, right? That minimizes then the amount of time that the patient is without immune protection. Right? And third, you have to think about this new immune system that's being rebooted. It's being rebooted based on, right, the genetics of the donor who's giving these stem cells. That means this new immune system that's being rebooted, right, is going to be geared up to follow different types of genetic variations or alleles that are present in the donor's genome, even though now you're expecting that new immune system to function in the recipient's body, therefore in the context of the recipient's genetic background. So therefore, the rebooted system must develop proper host tolerance. So let's look at this. So hematopoietic stem cell transplants, in these cases, usually the donor is going to be alive and healthy, right? And then they donate things like, stem cells. Right? So the patient, on the other hand, right, has to undergo a very arduous, very dangerous process. First, the patient receives cytotoxic drugs and radiation to sort of, in a blanket kind of way, destroy or erase their existing defective immune system. Right? That is really scary, right? Because it's one thing to say that your immune system is compromised or it's defective, right? But at least you still have it working, even if it's not working properly, right? To then, on purpose, completely erase your immune system to make way for a new one is a pretty horrifying thought, right? So that's why you're given strong chemotoxic drugs, right, that kill your immune cells throughout your body, and you're given high doses of radiation to try to destroy the existing bone marrow cells. Right? So we call this myeloablative therapy. So ablate means to destroy something, and in this case, you're destroying the myelin tissue of your bone marrow, where the original immune system grows from. Right? So you have to do that because you're going to be grafting in tissue from a donor. So you have to make sure that those donated stem cells don't get attacked by a pre-existing immune system. And another practical concern is you're actually trying to make room in the bone marrow so that the donated stem cells actually have the opportunity to be able to expand and actually create a new immune system within, right? So, I mean, these cartoons make it look so simple, right? Irradiation and chemotherapy to ablate or erase the existing immune system, then a transfusion of hematopoietic stem cells and hematopoietic tissues, right? Which then over time gets into all of the bones of the patient, and these stem cells begin to then grow, and you regenerate a new immune system in the space previously occupied by the old defective one. So transplanted tissue is usually done as a liquid. When you hear bone marrow transplant, I think most people think that somehow, the surgeons, right, are somehow scraping healthy bone marrow out of a donor's body and then somehow putting this bone marrow into the bones of the recipient. And that's kind of a weird thought, right? I mean, bone marrow, I don't know, sometimes like if you're at a fancy restaurant or something like that, right, they often serve bone marrow like as an appetizer or something like that. They give it to you in the bone, and then it's usually this very gelatinous kind of tissue in the middle. I mean, the truth is it is very yummy, right? But now that I'm putting it in the context of our lecture, it makes it sound really gross. But no, right? Bone marrow transplant is not you scooping out bone marrow at a fancy restaurant and putting it on your plate or something. No, it's actually a liquid, right? So these hematopoietic cells are taken, they're purified, they're cleaned up, and they're given to the recipient as an intravenous introduction, right? And therefore it flows, all these hematopoietic stem cells flow through the patient's circulatory system. And as they're flowing through capillaries that get into the bone, that's when these stem cells then hopefully come out of the blood into the tissue and make a new home there, right? So within 2 to 3 weeks of a successful stem cell transplant, you will begin to see things like immune cells, like the granulocytes, like mast cells, eosinophils, things like that, as well as natural killer cells begin to repopulate in the in the patient's body. Right? Keep in mind, right, these are cells that reflect the genetic makeup of the donor, right? So these new immune cells carry the genome of the donor, right, not the recipient. Right? B cells and T cells take a while longer, and I think you can probably understand why, right? Because we've learned all about the very involved process of B cells developing and the even more involved process of T cells developing, like in the thymus, right? So beyond 2 to 3 weeks, it probably takes a few more weeks before you see things like B cells and T cells being restored in the body. But again, right, this is the weird part. I mean, I think this is weird, right? The fact that all these new cells that are beginning to populate the body, right, are actually, right, cells with genomes in them from the donor, not the recipient, right? Because they're all from stem cells that came from the donated tissue. So what sorts of issues would you encounter now? Right? Well, it's quite possible that because these hematopoietic stem cell transplants are taking place over this IV, there's going to be all kinds of immune cells in the IV as well. So there might be T cells that come from the donor that also end up hitching a ride into the recipient. Right. And so these poor T cells, they don't know what the heck's going on, right? Because one moment they were in the original body that they were born in and doing their job in. Next thing you know, they've been extracted, right? And now they've been put into somebody else's arm, right? And they're flowing through the blood of this new person, right? And they're like, I have no idea what happened, but all right, I'm a T cell, so I better resume my T cell job. The problem is what? These T cells are looking all around them in the new body, right? And they're seeing lots of things that are different, right? Maybe not most of the proteins, because again, I said, right, most of the proteins that your body makes and that my body makes are going to be really similar, right? But certainly they're going to see things like, wait a minute, we are surrounded now by a different HLA type, right? So they may freak out, right? So this might trigger what we call a graft versus host disease, where the transplanted cells of the bone marrow itself, once it gets in the new host, they begin to go to work attacking like everything that they see, right? So this can cause a lot of inflammation to happen, right? But fortunately, the relative number of participating T cells that are involved here is pretty limited, because it's just whatever T cells were already like mature and working in the donor that hitched a ride, right? They're causing the problem in the host body, but they only last like a few weeks, right? Before they basically have to be recycled. And then once they disappear, that's the end of all the mature T cells that came over, right? And it can cause this problem. So that's okay, right? So that means the first few months of transplantation, the recipient has to take a lot of anti-inflammatory types of drugs to keep the inflammation down from this GVHD, right? Or graft versus host, right? Another issue having to do with the HLA matching is that hopefully you realize, right? You want to try to match HLA profiles, polymorphic profiles between donor and recipient as closely as possible, right? So that you don't get any kind of bad interaction, right? Think about this. The donor thymocytes, like the T cells, right, that come from the donor, right, they will be trained in the donor's body, right, to recognize and tolerate the donor's MHC or HLA type, right? So likewise, stem cells that are put from the donor into the recipient, as some of those stem cells begin to grow and start making things like thymocytes that then go to the patient's own thymus to actually become T cells, right, and make their T cell receptor, you're okay, because as they're making their new T cell receptors, they get trained against the host's MHC type, right, as they mature to become T cells, and then they go to work in the body, right? So let's look here. In this cartoon, slide 1, right, in the thymus, you had the transplanted stem cells made their way to the bone marrow. They successfully began to grow, and the stem cells begin to give rise to these precursor cells that then make it into the thymus, right, of the patient. And yes, this is wonderful, right? Because now suddenly you have now healthy T cells forming in the thymus that are being trained against the patient's own HLA types, right? That's why we see in this little cartoon, any of the T cell developing thymocytes that successfully, right, have just that right balance of recognizing the HLA so that you know that these T cells will work if they go to work in the body, but not bind it too tightly, right? The ones that maybe don't work are the ones that are just undergoing apoptosis in the middle. So these ones that do work, that are properly trained, they're released into the blood as the new generation of healthy T cells, right, as part of this rebooted immune system. And that's wonderful. And they are trained to recognize, right, and tolerate the patient's own HLA profile, because that's how they were taught or selected for in the thymus. But the problem is this, right? And this is kind of crazy and weird if you think about it. Remember, T cells depend a lot on other immune cells to show them things, right? And how do those other immune cells show them things in order for, let's say, a naive T cell to actually be activated, right? Dendritic cells, other professional antigen presenting cells show naive T cells different signals based upon the MHC that they carry, right? Because it's the MHC on these cells that carry small peptides that a naive T cell can check out for the first time, right? And you're like, yeah, okay, that's what we learned. But those professional antigen presenting cells, right, where did they come from? You're like, well, they came from the immune system, right? But wait a minute, which immune system, right? Because the patient's original immune system was wiped out. So these professional antigen presenting cells came from the rebooted immune system, right? Yeah, right, of course. The stem cells that gave rise to these new T cells also give rise to all these other professional antigen presenting cells. But the problem is, right, their version of the MHC comes from the stem cells, which comes from the donor. So that means all of the professional antigen presenting cells that are replenishing the new immune system are using the donor's MHC, right? And that's not what these brand new T cells have been raised and selected for in order for them to work. So as a result, you might have a problem here where even though it seems like the immune system has successfully rebooted, you're making T cells, you have plenty of dendritic cells, other types of professional antigen presenting cells, they can't talk to each other, right? Because the T cells that are being made don't recognize the MHC that the rebooted dendritic cells are using to show them. And as a result, it's kind of sad because in a way, even though the stem cell transplantation looked like it was a success, over time you realize, oh, it wasn't, because the person doesn't really have a strong adaptive immune system anymore, right? Okay. So here are some interesting things to consider also. When I described this graft versus host disease, right, I said the transplanted immune cells might actually attack the host. And I made it sound like that's like a super bad thing, even though it might be kind of tolerable, because after those transplanted cells have sort of gone their way, right, then maybe the problem's gone. But the interesting thing is sometimes a little bit of graft versus host disease can be useful. Useful if you're dealing with something like cancer. So, in cancer, when a tumor, let's say, is successfully removed via surgery, maybe with a combination of chemotherapy, radiation therapy, there's always the fear that there's going to be some cancer cells left over, right? And those cancer cells then, over time, can be the start of forming new tumors and a new relapse of the cancer, right? So wouldn't it be great if you're dealing with a hematopoietic cancer, let's say a leukemia or a lymphoma, that involves having to erase the old immune system, right, and put in a new immune system? Actually, wouldn't it be nice if that new immune system could in some way also see the old cancer cells that are left over and attack them and sort of really clear them out, right? So that involves the grafted tissue somehow attacking some component of the host. Right? So in this case, let's look at this. Let's pretend you have a family, right? And you have the mother and the father, right? And the mother and the father each carry their own set of different polymorphisms. So let us pretend, for example, that this shape right here represents a section of the chromosome where the different HLA proteins, genes are located. So this is your MHC complex, right? And mom would have one version that maybe she got from her dad and one version that she got from her mom, right? So mom in this case carries the blue and the green. Dad in this case carries pink and yellow, right? And each one of these little blocks here represents a different one of those HLA proteins, A, B, C, and the different HLA-D ones that make up MHC1 and MHC2. So if they go ahead and have kids, so here they are over here in the context of their family. Here's mom, right? She carries haplotype 1 and 2. Dad carries haplotype 3 and 4. Oh, and remember, I say haplotype because remember, when we are passing our genes on to our children, right, we do so through the process of meiosis, that are gamete-forming cells in our body, the cells that give rise to egg and the cells that give rise to sperm, right? They undergo meiosis so that you have some crossing over potential, the mixing and matching of different genes from the grandparents, right? But remember, genes, when that happens, the crossing over, it means the physical crossing over of homologous chromosomes, right? So two of the same type of chromosome during meiosis one, they have the ability to cross over and actually exchange pieces of the chromosome. So it's not like individual genes are going over, right? It's actually chunks of the chromosome. So that means blocks of genes. Whatever genes are part of that section of the DNA that gets swapped over is what gets mixed up, right? So we call that a haplotype block, a block of genes that, because of their physical closeness to each other on the same chromosome, will tend to move or be swapped out during meiosis together, right? So the MHC genes are like that, right? Because they're all part of the same MHC complex. So they will move as this haplotype block of genes, right? So that's why we can represent this as these colors: blue, green, pink, and yellow, right? So these parents, they have 4 kids here, right? So that, of course, each child, right, would have one from mom and one from dad, right? So in this case, blue and pink, in this case, green and yellow, right? Let's say this sibling here is the one that gets sick, right, and develops some sort of hematopoietic cancer, right? So because this individual, this girl here, right, she has the blue haplotype 1 and the yellow haplotype 4, right? That means in order to do a good job of matching HLA types, this sister and this brother could be donors, right? Because in either one of these cases, they at least have one of these haplotypes. This brother over here would not be a suitable donor, right? Because he has the green and pink ones that he got from mom and dad, right? Which his sister over here, the sister who's sick and in need of the transplantation, right? She doesn't have that, right? So oftentimes, in the case of cancers like this, they will on purpose be okay with using a haplo-identical transplant, meaning having only half of the haplotype profile matching. Why? So that there's a chance for the new rebooted immune system to still see pre-existing leftover cancer cells of a different haplotype and being able to actually attack them. So let's say here, in this case, the donor that was chosen has the blue and the green haplotypes. The recipient, maybe it was the sister, right? She's green and pink. So they on purpose go ahead and do this so that the donor's hematopoietic cells, right? So these are the cells that are now have been transplanted over and are trying to reboot the system, right? All of the immune cells that these stem cells make, including things like the natural killer cells, have this same blue and green, right? So that means natural killer cells are trained. Hey, you are all about being okay with blue and green. When you go to work, right, and you encounter other cells, if they are blue and green, you know they're good, right? So when they go to work then, as they're going through the body, what do they encounter? They may encounter some of the old leftover leukemia cells, right, that came from the girl's original immune system. Remember on the left, she was born as green and pink, right? So this immune cell from her old immune system that was cancerous, right, is is green and pink. Therefore, the natural killer cells, they're okay with the fact that they see the green part, right? So green signals, right, represented here on the green MHC, right? But they're not okay with the fact that the cell also expresses the pink version here, right? So as a result, the natural killer cells will say, there's something weird going on here. So they might kill this cancer cell that was left over. If they didn't kill this cancer cell, maybe this cancer cell would start dividing again, right? And result in the relapse of the cancer. So this particular type of strategy is actually used, right? So we call this graft versus leukemia effect. And it's used very successfully, right? To be able to clear the remaining leftover cancer cells out of a leukemia patient as part of rebooting the new immune system from the stem cell transplant. Okay. So more and more, I think surgeons are recognizing the usefulness of sort of playing one immune system against another, right? And being able to have different HLA types be tolerant, right? And accepting. This is an interesting phenomenon. Around 8% of non-identical twins, and non-identical, meaning that you had a situation where there were, let's say, 2 eggs present at the time of fertilization, and both eggs got fertilized, right? So you end up with 2 babies forming, and genetically, right, they are different. They are not identical twins because they are 2 different eggs to start with, right? And those eggs would be different from each other because each egg is a result of different meiosis recombining things, and of course, different sperm cells, which each one of those sperm cells are different because they are the product of different meiotic crossing over in dad, right? So as a result, you get two kids who are genetically not identical, but they are, what we call non-identical twins, right? But because they are developing at the same time in the same womb, right? Oftentimes they share a lot of their blood. So as a result, such siblings in utero are exposed to each other's non-self HLA types. Right? So as part of the way their immune systems are starting to kind of develop, the ability to tolerate this other non-self HLA type becomes sort of part of how this immune system learns to sort of function. Right? And what that means is that much later in life, for example, as adults, right, these non-identical twins, right, are very good at being good donors for each other, organ transplants, because their immune systems, right, from the very start were tolerant of this other person's HLA type. Right? And so it's possible now that many surgeons who are doing transplants, what they're trying to do now is at the same time that you're transplanting something like a kidney, right, you also try to transplant a little bit of hematopoietic stem cell tissue to, in a way, allow the host's immune system to kind of have a glimpse, right, at another immune system, another sort of set of HLAs that are coming from immune cells to hopefully sort of lessen or dampen the allo reaction that might otherwise take place. And the hope is by doing so, you will lessen the need for the transplant patient to take such heavy doses of immunosuppressive drugs. Okay. Very interesting to think about how the more we learn about how our immune systems, right, sort of deal with other types of immune systems, somebody else's immune system, the more we can hopefully leverage that to try to overcome these challenges of doing things like tissue transplantation. Okay, let's go ahead and stop there.